All right, today we're going to dive into one of the most elegant, most critical events in all of embryonic development, neurulation. We're going to watch how a simple sheet of cells folds, fuses, and splits to create the entire foundation of our nervous system. It is a true masterclass in biological engineering. So here's our game plan. We'll start with the initial setup inside the embryo, then we'll get into the signals and the mechanics that drive this incredible transformation from a flat plate into a tube. And finally, we'll see how this one single event gives rise to two completely distinct systems, each with a very different job to do. So, where does our story begin? Well, it kicks off right after a process called gastrulation. Now, at this point, the embryo is no longer just a simple ball of cells. It's organized itself into three distinct layers, what we call the primary germ layers. This is the absolute foundation upon which every organ and tissue will be built. Okay, so you've got these three layers, right? But for our story today, the one we're going to zero in on is that top layer, the ectoderm. You can think of it as the exterior layer. It's going to become our skin, our interface with the outside world, and pretty amazingly, it's also going to form the entire nervous system that interprets that world. So you might be asking, how does the embryo decide which part of the ectoderm becomes the nervous system? It's not random. It all comes down to a signal. The layer sitting right underneath, the mesoderm, starts sending out these chemical messages. It's kind of like it taps the ectoderm cells right above it on the shoulder and says, hey, you've got a new job. You're going to become neural tissue. And that newly specified region, we call that the neuroectoderm. Okay, so these cells have got their marching orders. Now what? The next big step is all about organization. How do they actually assemble themselves into the very first structure of the nervous system? What happens is these newly designated cells start to change shape. They get taller, more columnar, and they pack together really, really tightly. This whole process thickens the area into this distinct, flat sheet, and that is the neural plate. You can literally think of this as the foundation, the architectural blueprint from which the entire brain and spinal cord will be built. And this right here, this diagram, just perfectly captures that initial state. You can see the flat neural plate right there in the center. But, and this is key, pay close attention to the cells at the very edges, kind of like the shoulders of the plate. Those are the prospective neural crest cells. Right now, they're part of the team, but they are destined for a totally different journey. Keep an eye on them. Now, a flat plate is great, but development is all about movement and change. So the million-dollar question is, how does this simple, two-dimensional sheet pull off one of the most profound transformations in all of biology and become a three-dimensional tube? Well, that whole process has a name. It's called neurulation. And it's basically this perfectly choreographed ballet of cell shape changes and movements that rolls the flat plate up into a hollow tube. This neural tube is the primordium, the very earliest stage of what's going to become the brain and spinal cord. And check this out. You can see it happening right here. Look at diagram B. The midline of the plate starts to buckle downwards, creating what's called the neural groove. And as that happens, the outer edges, the neural folds, start to rise up. It's almost like two waves gathering to crash together. And this isn't just some random collapse. It's a highly coordinated movement. The folds keep pushing toward the midline, and honestly, the zipper analogy is perfect here. The tube doesn't just fuse all at once. Nope. It starts zipping up right in the middle of the embryo and then closes simultaneously towards the head and the tail. And this diagram shows the climax of the whole fold. The very tips of those neural folds meet and fuse together at the midline. In this one single efficient moment, two incredibly important things happen. First, you've created a self-contained hollow neural tube internally. And second, it pinches off completely from the surface ectoderm, which now just seals over the top to become the future skin. Now, remember that zipper analogy? As it's closing towards both ends of the embryo, it leaves two little openings for a short time, the anterior neuropore at the future head and the posterior neuropore down at the tail. And the timing for when those finally close, it is absolutely critical, as we're about to see. But wait, the story isn't just about the tube. At the exact moment of that fusion, something else extraordinary happens. Right there at the seam where the folds meet, a completely distinct population of cells is born. And look at this. This is the beautiful final arrangement in cross-section. You've got the internalized neural tube, the surface ectoderm has healed over the top, and nestled right there between them are those cells we flagged earlier, the neural crest. Honestly, these neural crest cells are one of the true marvels of development. As those folds fuse, they undergo a process called delamination. They literally break away from the sheet. They go from being stationary parts of a structure to being these free-roaming migratory cells. And they're also multipotent, which means a single one of these cells has the potential to become tons of different cell types. So this table really lays out the fundamental split, right? From just one sheet of cells, the embryo has brilliantly created two populations with completely opposite job descriptions. One, the neural tube, is stationary and structured. It's going to be the central processor, the CNS. The other, the neural crest, is migratory and versatile. It's going to travel out and form the network cables, the PNS. Okay, so we've got our two starting populations. What do they ultimately become? Let's take a look at the final destinies of the tube versus the crest.
For the neural tube, its destiny is pretty clear-cut. It becomes the entire central nervous system. The front end balloons out and folds into the complex architecture of the brain, while the back end elongates to form the spinal cord. And the inner lining of this tube is just packed with progenitor cells. Think of them as dedicated stem cells that are going to generate every single neuron and support cell in your brain and spinal cord.
And while all that's happening, those intrepid neural crest cells are on the move. They migrate along these complex pathways to almost every part of the body. Their mission is to build the entire peripheral nervous system, the sensory nerves that let us feel, the autonomic nerves that control our organs without us thinking about it, and so much more. It's just an astonishing diversification from a single starting population.
And this question brings the whole process into sharp clinical focus. Because for all its elegance, the process of neurulation is incredibly vulnerable. Even tiny errors in the timing or the mechanics of this fold can have absolutely devastating consequences.